Now next we are going to see the ultrastructure of a nephron. As you know, nephron is the functional unit of a kidney. There are more than 1 million nephrons that are present inside a kidney, each kidney. So these are the functional units of kidney. They are the main performing areas where the urine formation would take place, because the main function of kidney is getting rid of the nitrogenous waste in the form of urea, and that is given out in the form of urine. So we are going to study about the functional unit of kidney now. If we have to analyze the structure of nephron, it can be divided into two parts. First one is glomerulus and the second one is renal tubule. Now the renal word is denoted for kidney, so there would be a tubular structure that is present inside the kidney. Now recall your knowledge about the structure of kidney, that it was divided into outer cortex and inner medulla. So supposedly we are representing this kidney in the form of cortex and medulla, and cortex ends somewhat over here. We have medulla over here. We are showing the structure of a nephron over here. Okay, now we are going to see what all is present inside the nephron. Where is glomerulus, where is the renal tubule, the two main parts which are the clear-cut distinction present in the nephron. So we have glomerulus, that is the tuft of capillaries. This red color I have used to represent the arteries, basically the capillary system that is present inside the nephron. You know that because the filtration has to take place from the blood itself, so there should be an ample supply of capillary system within the nephron or within the kidney. So we have a tuft of capillaries which is enclosed inside the Bowman's capsule. So this part, this tuft of capillaries is glomerulus. Okay. Now this glomerulus is enclosed within the tubular part that is known as Bowman's capsule. Or I would rephrase it that the glomerulus is encapsulated between a tubular part known as Bowman's capsule. Over here, this particular part, this entire part, glomerulus and Bowman's capsule, this part is known as renal corpuscle. So over here we are done with the glomerulus part, and the remaining part of the nephron is known as the renal tubule, which extends till medulla of the kidney. Cortex is outer side and it goes inside the kidney. Now this Bowman's capsule, it opens up into a highly convoluted tube-like structure that is known as proximal convoluted tubule. Proximal because it lies close to the glomerulus, or in short we call it PCT. Then this PCT, basically the absorptive part, it is absorptive and secretory part. Absorption has started taking place from the proximal convoluted tubule itself. If we have to analyze the mechanism of urine formation, this proximal convoluted tubule leads to a hairpin like structure. You know, the hairpin structure is like this. So this U-shaped structure is known as loop of Henle, or we call it Henle's loop. This loop of Henle can be divided into two further parts. This limb, there are two limbs of loop of Henle because it is a U-shaped structure. So this one from which is joined, this limb which is joined to the proximal convoluted tubule is known as descending limb. Now why it is named as descending limb, there is a reason, because the filtrate which has come from the arterioles, it comes and descends in this particular limb. That means it goes downwards like this, and after it has reached over here, it has to go upwards according to the gradient, and the absorptive tendencies of both the sides are different. Alright, they are lined by an epithelium. See, this is a tube-like structure that is lined by epithelium which would absorb the maximum of blood, has given out the content of it which is equivalent to plasma. Only proteins are left inside, and of course RBCs and WBCs. Rest of the content of the blood is being dropped into the glomerulus. It comes from this way and it has to reach the collecting duct. Okay, so this passage basically is the passage that the filtrate takes, and by the time it reaches till here, that is the collecting duct, this part is known as collecting duct. By the time it reaches the collecting duct, it has become urine. Okay, so what all is left inside that there would be some watery content, so that urea does not burn the bladder, and the rest of nitrogenous waste that would be left. But in the beginning there is lot of water content that is present, so maximum of reabsorption takes place in this loop of Henle. We had reached till descending limb, so would be the ascending limb, where the flow of filtrate would be opposite to that present in the descending limb. So as you can see, it is having a greater area inside it, the tubular radius is greater, then it becomes thinner and thinner, then it takes a U-turn, it reaches distal convoluted tubule, because it lies distal to the glomerulus, so we have distal convoluted tubule. Again, this part is highly reabsorptive, and it is secretory as well. It secretes a few ions in order to maintain the ionic balance and takes up whatever is present. So we get that this part is for reabsorption of sodium chloride and water. This part, proximal and distal, these two convoluted tubules are for absorption of various ions and amino acids which are present, which are not to be wasted. And all these DCTs of different nephrons come and join in the collecting duct, which is the site where urine is present. These collecting ducts would open up into the ureter. Many collecting ducts through calyces, they would open up into the ureter, and from there the urine would be thrown into the urinary bladder. So this is the ultrastructure of nephron that we need to understand. There are few things which are to be remembered, that this is the functional unit of kidney, that is very clear. There are more than 1 million nephrons present in each kidney. And if we have to see the structure of nephron, it is divided into two parts. First one is glomerulus, that is the tuft of capillaries, and then there is the renal tubule. Now I have represented this reddish part. I told you this is the representation of the blood supply, and this particular capillary tuft is known as vasa recta. All right. Now remember this name. Vasa recta is the capillary meshwork which surrounds the nephron, the tubules as well as the loop of Henle. This is the capillary network which is responsible for counter current mechanism. When we talk about the formation of urine and its concentration, we come across the counter current mechanism. I would just put a some light over it, that what counter current mechanism would be all about. Just from the structure you can make it out that in this descending limb, the urine is descending down while it is ascending in the ascending limb, but it is opposite in the case of the vasa recta, because you can see that this particular part of the capillary network is afferent arteriole. You have to remember, when some part enters inside the organ or the part, it is known as afferent, and when it leaves, the term efferent is used. Okay, so this is afferent arteriole, this is the efferent arteriole. This efferent arteriole, after the filtration, once the filtration slits have got rid of the ultrafiltrate that is to be thrown into this PCT, they would take the remaining blood and it would enter the vasa recta. As you can see, this part, this glomerular part, it had an afferent arteriole. That afferent arteriole brought the blood, it took place and the ultrafiltration took place. The urine was thrown, not urine exactly, the ultrafiltrate was thrown into the PCT, and the remaining blood, it came out from the efferent arteriole, and again it convoluted itself around the loop of Henle. Now this blood, which is devoid of the water, maximum water content, it would take up the water back from this loop of Henle. Alright. Now this capillary network has an opposite flow of blood. Like supposedly over here you have the downward flow of the ultrafiltrate, the flow of blood would be upwards in this particular vasa recta part, and in this part it would be downwards, so that whatever water is being reabsorbed, it can be reabsorbed in the blood, and that blood that goes out is not devoid of water, it has only got rid of the nitrogenous waste that it had to remove. Now one more thing you have to remember, that this loop of Henle and vasa recta go hand in hand, and wherever this loop of Henle is quite longer, as it goes deeper into the medulla, the organism would be able to reabsorb water more. As in the case of camels and kangaroo rats, such animals, they are able to absorb maximum amount of water and they can survive without water. That is due to this longer loop of Henle, which is able to reabsorb more water in their blood, so that minimum water loss is taken up. And you have to remember that it is the capillary system and its counter current mechanism which is responsible for taking up the water which has been reabsorbed from the loop of Henle. The permeability to different substances varies, that we study in the mechanism of urine formation. For now, you have understood the structure and you have to remember all these parts. I am repeating it again, that there is a renal corpuscle that comprises of glomerulus and Bowman's capsule. There is afferent arteriole that enters the glomerulus, efferent that comes out, sorry, and then this efferent arteriole, it forms a vasa recta network. Recta basically stands for network. So this is vascular network which surrounds the renal tubule part of the nephron. What is in renal tubule? There is PCT, there is DCT, and in between there is a hairpin loop like structure, that is loop of Henle. And this DCT, that is distal convoluted tubule, after remaining absorption which was not completed till here, it throws whatever waste is present inside the collecting duct, and collecting duct is also responsible for absorbing maximum water. So what is left while the collecting duct ends up into the calyces is the urine that would be thrown into the ureters and then into urinary bladder.